Good day. Lecture about Corynebacterium, causative agent of diphtheria. Diphtheria is an acute, toxic-mediated disease caused by toxigenic Corynebacterium diphtheria. It's a very contagious and potentially life-threatening bacterial disease. It is localized infection disease which usually attach the throat, nose, mucous membrane, and another cells. Taxonomy: Kingdom Bacteria, Phylum Firmicutes, Family Corynebacteriaceae, Genus Corynebacterium, Species Corynebacterium diphtheria, Biotype Gravis intermedius and metis, and species Diphtheroid Coryneiform, Corynebacteria pseudodiphtericum, Corynebacteria xerosis, and Corynebacteria ulcerans. This microorganism cause another disease. Morphology: they are thin, slender gram-positive bacilli, but are easily decoralized, particularly in old cultures. They have a tendency to clubbing at the one or both ends. They are highly pleomorphic. They are non-motile, non-spore-forming, and contain metachromatic granules, volutin granules, or bulbous earnest granules. The bacilli are arranged in a characteristic fashion and smear. They are usually seen in pairs, palisades, resembling stakes of the fence, or small groups or as individual cells lying at sharp angles to another, resembling the letters V or L. For microscopic examination, we use gram staining technique. It's gram-positive microorganism and Corynebacteria for study. For staining Corynebacteria diphtheria, we use specific staining technique because Corynebacteria includes granules, and we use Albert Neisser staining technique and Loeffler's method staining. Albert stain slides showing green color bacilli body and brown or dark purple color metachromatic granule, you see. Neisser stain slides showing yellow color bacillary body and brown or dark purple color of mitochondrial granules. And Loeffler's method, we use methylene blue slide showing blue color bacilli body and brown or dark purple color metachromatic granules. Cultural characteristics: Corynebacteria diphtheria is an aerobe and facultative anaerobe. The optimum temperature for growth is 37 degrees, range 15 to 14 degrees, and optimum pH 7.2. Corynebacteria diphtheria don't grow on surface simple nutrient media, and will need complex nutrient media complex nutrient media for cultivation of Corynebacteria. For this nutrient media, it's Loeffler serum, telluride blood agar. The addition of potassium telluride makes the medium selective for Corynebacteria by inhibiting most other pathogenic and commensal bacteria on this medium. Corynebacteria diphtheria give gray, black, shiny or dull black colony. Reduce the telluride salt. We used Tinsdale's medium, the addition of cystine to a telluride containing medium. We use Columbia blood agar and Klauberg agar for cultivation of Corynebacteria diphtheria. On this slide you can see Loeffler serum for Corynebacterium diphtheria. Colony are at first small, circular, white, opaque disc, but enlarge on continued incubation and may acquire a distinct yellow tint. In 18 hours, the Löffler's length yield organism of typical diphtheria-like morphology. And if we will study pure culture of Corynebacteria diphtheria under microscope. On this slide you can see telluride blood agar, colony Corynebacterium on surface telluride blood agar, black colony, Klauberg agar, black colony, you see Tinsdale agar, black colony, because Corynebacteria reduce telluride salt and reduce cystine on surface Tinsdale agar. But first slide I explained about biotype of Corynebacteria diphtheria, which cause diphtheria disease, and Corynebacteria diphtheria gravis, Corynebacteria diphtheria intermedius, and Corynebacteria metis have different properties, and we can investigate these properties by morphology, characteristic colony on telluride blood agar, consistency of colonies, hemolytic activity, different growth in broth, glycogen and starch fermentation, toxigenic strains, virulence, and predominant strains. Different characteristics I will not read, we will study, I will ask you in our laboratory class. For identification, we use a biochemical reaction, and we use nitrate test, urease activity. We will investigate catalase production, testinase activity, pyrazinamidase activity, carbohydrolate fermentation. We use this biochemical reaction for identification Corynebacteria diphtheria, different biovars, and with another coryneiforms microorganism, means Corynebacterium pseudodiphtericum, xerosis, and ulcerans. For study toxigenicity, we will use this toxigenic infection, and for study produce Corynebacteria diphtheria or don't produce Corynebacteria diphtheria toxin, we will use Alex gel precipitation test. On this slide you can see, for example, reaction, positive and negative reaction. Principle of Alex test. This Alex plate demonstrates the toxigenicity of Corynebacterium diphtheria. Toxins produced by the bacteria growth will diffuse in agar, and where it meets the antitoxin at optimal concentration will produce a line of precipitation. A negative control should be free of any line. No precipitate will form in case of non-toxigenic strains. On this slide you can see Alex test toxigenic strains, positive reaction, negative reaction, and control. Antigenic structure. O antigen group lipopolysaccharide cell wall thermostable antigens gives cross reaction with antigen of mycobacteria. Surface protein K antigens has immunogenicity, provides specificity, thermal label. K antigen presented capsular nucleoproteins. A deep seed antigen found in all Corynebacterial species as well as in Mycobacterium tuberculosis. Based on antigenic structure, classified Corynebacteria diphtheria into 58 serotypes. Diphtheria metis includes 14 serotype, diphtheria gravis 14 serovariant, and diphtheria intermedius 4 serovariation. Bacteriophage typing, Corynebacteria classified by 15 by 15 types. Factors of pathogenicity or virulence factors. Factors of adhesion. Polysaccharide and lipids of the cell wall, lipoteichoic acid and other structure which may be on the surface of microbe, capsules and a cell wall. Factors of invasion. Fibrinolysin, neuraminidase, hyaluronidase, superficial toxic glycoprotein, dimethylate triglycerol. Factors of adhesion, microcapsule, lipids of cell wall, catalase, neuraminidase, DNase. Factors of toxin formation, exotoxin production only in infected by virus phage infected carrying toxin gene. Toxigenic strains of Corynebacteria diphtheria produces a very powerful exotoxin. The toxicity observed in diphtheria is directly attributed to the toxin secreted by the bacteria at the site of infection. Diphtheria toxin heat-label protein. It is extremely potent and lethal for human. The toxin consists of two fragments, A, active, and B, bonding. Both fragments are necessary for the toxic effect. Fragment A has all the enzymatic activity, whereas fragment B is responsible for bonding the toxins to cells. Fragment A is responsible for the for the toxigenicity. Fragment B bind to the receptors on the eukaryotic cells and mediate the entry of fragment A. In the presence of nicotinamide adenine dinucleotide, fragment A inactivates the elongation factors EF2. Therefore, inhibition of polypeptide chain elongation, leading to inhibition of protein synthesis, and this causes cell death. Toxin has special affinity for myocardium, adrenals, nerve ending. On this slide you can see mechanism action of diphtheria toxin to subunit A, active subunit B, bonding subunit. B component of the toxin attach to host cell receptors and give possible subunit A entry into the host cells. Subunit A inactivates elongation factor which form polypeptide chain of proteins and block synthesized proteins by ribosomes and cause death of cells. Epidemiology. Portal to entry, skin, respiratory tract, cuts and wounds. Source of infection, sick person or carrier, convalescence or health. Of toxigenic strains of Corynebacteria diphtheria. Ways of transmission, airborne, contact, household, case, rate, sporadic, outbreak are possible. Immunodeficiency, antitoxic, post-vaccine. Seasonality, autumn, winter. Diphtheria is a toxemia because main factors pathogenicity which can kill your patient, can death of your patient. It's toxin. I repeat, toxin. It's histotoxin which cause block, elongation, block formation of protein on surface ribosome. Main factors pathogenicity for your patient is toxin. Diphtheria is spread, transmitted from person to person, usually through respiratory droplets, like from coughing or sneezing. Rarely, people can get sick from touching open sores, skin lesions, or close the touched open sores of some sick with diphtheria. A person also can get diphtheria by coming in contact with an object, like a toy, that has the bacteria that cause diphtheria on it. In the upper respiratory tract, diphtheria bacilli elicit an inflammatory exudate and cause necrosis of the cells. The diphtheria toxin possibly assists colonization of throat or skin by killing epithelial cells or neutrophils. The organism do not penetrate deeply into the mucosal tissue, and bacteremia does not usually occur. The exotoxin is produced locally and spread by the bloodstream to distant organs, with a special affinity to hurt muscles, the peripheral nervous systems, and the adrenal glands. Laboratory diagnosis of diphtheria. Laboratory diagnosis consists of isolation of diphtheria bacilli and demonstration of its toxicity. ELEC test we use for study toxigenic properties of Corynebacteria diphtheria. Detection of the antigen and smear from a throat and nose, taking of material on border between affected area and normal mucus. Microscopy we use special staining technique, Neisser, Albert staining technique, Loeffler staining technique. We will see typical locating of rods, grains of volutin in bacteria. Bacteriological method. We use complex nutrient media and we isolation of pure cultural, and after that study colony microorganisms, study morphological features, and for identification we use biochemical reaction and we use ELEC test for study toxigenicity, because just Corynebacteria diphtheria, which includes three biotype, gravis, medius, and metis and intermedius, produce toxin. Another strains of Corynebacteria do not produce toxin. We use serological test for detection of antigens and antibody, ELISA, reaction immunofluorescence, and another test. We use PCR technique for detection of genetic material of bacteria. We use in 1913 years we use Schick test, introduced an intradermal test for distinguish between susceptible and immune persons. And now we don't use because laboratory technique give full information about biochemical, morphological, and antigenic properties of Corynebacteria diphtheria. And in vivo subcutaneous test and intracutaneous test we used for laboratory diagnosis Corynebacterium diphtheria. Prophylactics. The method of immunization available are active and passive or combined method for prophylaxis diphtheria disease. For active prophylaxis we use vaccine DTP, diphtheria, tetanus, pertussis vaccine, or DTAP. We used TD vaccine, diphtheria, tetanus toxoid, and DTAP. Diphtheria, I repeat, diphtheria is toxinemic disease. And for prevention and for treatment your patient, we will use toxoid. Toxoid is anti-toxin effect upon toxin of bacteria. And passive prophylaxis, this is emergency measure to be employed where susceptible are exposed to infection, as when a cause diphtheria is admitted to general pediatric wards. It consists of subcutaneous administration of 1,000 units of antitoxin antidiphtheriacum serum. Schedule of primary immunization. DTaP for young children 2, 4, and 6 months, 15 through 18 months, 4 through 6 years. For preteens, 11 through 12 years. For TD or TDAP vaccine for adults, we will use vaccine every 10 years. Treatment. First, hospitalization. Specific treatment. Introducing of antitoxic antidiphtherial serum, because I repeat, main factors pathogenicity of Corynebacteria diphtheria is their toxin. We use glucocorticoids, antibiotics. In case of group, inhalation, bronchiolitis, and another symptomatic treatment. But don't forget, antitoxin serum we use for treatment your patient. Thank you for your attention. Don't forget, take the computer test in your personal account.